I would describe my research with these three words: antibodies, protein design, and neurodegeneration. I hope this research can help in the development of treatments against devastating diseases in the future by replacing costly in vivo methods with computational designs. Naturally, proteins exist in a mixture in cells. However, when we want to study the function of proteins, we are usually only interested in one of them. So, protein chromatography uses different kinds of materials which are packed into a column, and the proteins interact more or less strongly with this material. By flowing the mixture through the column, the proteins are therefore retained for more or less time on the column, and they are therefore separated, and we can obtain them in a pure state. From the network, I would probably like to work here at Lens. They perform very interesting research at the intersection of optics and biology, so I think this would be a very interesting opportunity to expand my skill set.